Okay, so it's 9 o'clock. Hello, everyone. Welcome to our today's online museum seminar. It's absolutely my great pleasure to welcome my colleague and then friend, Dr. Jakob Moltke from Seattle. Jakob currently is associate professor of immunology in Washington University. So very brief introduction about Jakob. He completed his undergraduate study in Dartmouth College in genetics and cell and development biology, and then continue with the training in Columbia and UC Berkeley in biotechnology and molecular cell biology. So after he complete his PhD in 2012. He joined the lab in UCSF as a postdoc in infection disease. So from 2016, after he completed his postdoc training, he started his independent research program in Department of Immunology, University of Washington in Seattle. And then 2 years ago, 2022, he was promoted as an associate professor in University of Washington. So Jacob's lab is particularly interested in innate immune sensing and type 2, in particular allergic immunity. His lab studied the initiation of type 2 immune response by parasites, allergens, with a current focus on the role of epithelial tuft cells, which is a quite unique epithelial subset during the process. He actually was the very first one of the scientists to study the link between the tuft cells to the type 2 immunity, and also discovery protist-derived assassinate as the first intestinal tuft cell ligand for tuft cell effector function. His lab also contributed to many studies of tuft cells in the thymus and the trachea. As such, he helped to define the rapid emergent field studying tuft cells immunity, which I think a lot of currently trainee and also the audience probably also interested. So the ongoing project for Jacob's lab, including further discovery of innate immune sensing machinery in type 2 immunity, identification of additional tuft cell effective functions, and the characterization of tuft cell contribution to adopt immunity. His lab actually cover several aspects of epithelial biology, which including tuft cell and innate immune sensing of intestinal helminths and process, and then also to generate a new model for activation of group 2 innate lymphoid cells. And then he's also interested in discovering and characterization of cytokine-independent inflammasome activity. So both his postdoc and after he become independent, Jakob is very productive and then generate a lot of groundbreaking discoveries. With these contribution to the scientific community, he was awarded in numerous honors, which I'm not going to list all of them, but a few of them, including Young Investigator Presentation Awards, Pfizer Awards, American Association of Immunology, and he also was elected as a serial scholar. 2021, he was awarded as Bone Source Welcome Investigator at the Pathogenesis of Disease and the Regeneral New Investigator Awards in International Cytokine and Interferon Society. So I think today, Jakob, we're going to still tell us about his main work in tuft cells in the GI. And then very exciting to look to your presentation. Thank you so much, Jakob. Please go ahead.
Okay, thank you so much, John, for the invitation and for the introduction. Yeah, you've laid the groundwork for where I'm going with this. Just to reiterate that the main interest of the lab really is these type 2 immune responses that are initiated by both parasitic worms and allergens, right? So just an astonishingly diverse group of triggers that all feed into the same immune response. And we know how they start and we know how they end. We know the clinical symptoms of allergy, we know the cells that drive it, we know the cytokines that drive it. And more recently, we've discovered these innate lymphoid cells that sit in the tissue and are some of the first that get activated. But it's really how you detect these agonists in the first place, and then how it feeds into the immune response remains less well understood. And the epithelial barrier is undoubtedly a really important part of that. And we have focused on these tuft cells, which I'll explain to you how we ended up studying them. So the plan here today is quickly just tell you a little bit about tuft cells and the history of their study, because I know that they're not a cell that is probably that familiar to a lot of you. Then I'll tell you a project that we've just finished up on looking at the role of tuft cells and how they regulate epithelial physiology. And then at the end, sort of briefly, I'll touch on some newer direction having to do with tuft cells and adaptive immunity. So, right, tuft cells are these rare epithelial cells. They're found in many mucosal barriers. That includes parts of the airway, the upper airway, not the lower lung, but the trachea and the nasal cavity. Much of the intestinal tract, the bile duct, the gallbladder, the stomach, the small and large intestine, also in the urethra. And then additionally in the thymus, which is a little bit of an outlier from the other locations. And they were actually discovered more than 60 years ago, because when people started doing electron microscopy, they quickly saw that some of the cells in the lining of the epithelium looked different. And they were defined really by what was called the tuft, and this is why they're tuft cells. It has this protrusion at the top that looks like a little hairdo, and you can see by the scanning EM the way that these cells protrude up out of the border of the intestinal lining. So looking down onto the lining here. This is a tuft cell in the middle. Okay, so for many years, people took really nice pictures of the tuft cells, sort of uncovered all sorts of intriguing cell biology, but functionally they remained really mysterious for a long, long time. The first clues to their function came in the late 90s and early 2000s, and it actually came from the people who were trying to understand taste transduction. And they realized that all the tools they were building, like the antibodies and the reporter mice, we're also labeling these rare cells in the lining of the intestine, for example, or the airway, and that these were tuft cells. And so actually the first sort of transcriptome of tuft cells came out around this time and revealed that they really have very similar transcriptional programs as taste receptor cells. Then a little bit after that, or during that, is when their function first became clear. So this was all work done in the airways by really pioneering researchers who realized that there was some sensing of bacteria-derived molecules, and that the tuft cells could signal to neurons in the airways. And they also around this time discovered that POU2F3, which is the master transcription factor for type 2 taste cells, is also absolutely required for the differentiation of tuft cells. So mice that lack this have neither the ability to taste bitter, sweet, and umami, nor do they have any tuft cells. But despite all of that, the function of intestinal tuft cells remained a complete mystery. And I really stumbled into studying this when I was a postdoc, again, because I was interested in type 2 immunity and how it was being initiated. And I joined Richard Lox's lab at UCSF that was using this Nippostronzos brasiliensis hookworm model, and they had found these ILC2s recently. And we knew that one of the signals that activates ILC2s is this cytokine IL-25. And work that had been done previously revealed that if mice lack this cytokine, they have a big problem clearing worms out of the intestine. So a wild type mouse will clear them by day 10, but not these knockouts. What we didn't know was where IL-25 was coming from and how it was being regulated. And so we made a reporter mouse that we called Flare25, and we just inserted RFP downstream of the coding sequence for IL-25 and we went looking for red cells. And we found them in lots of barriers and up and down the intestinal tract in particular. So here you can see green is marking the epithelial lining of the small intestine. And here in red is one of these rare IL-25 positive cells. And so in work that I did at the time, we discovered that every one of these IL-25 positive cells is a tuft cell. And the only IL-25 positive cells we ever found were tuft cells. And all tuft cells express IL-25 as part of their transcriptional program. So it's really, at least in this reporter mouse, it's a one-to-one relationship between tuft cells and IL-25 in multiple tissues. The other thing that we found that was really exciting to us was that if you compare the number of tuft cells in an uninfected small intestine on the left here, they're rare. This is 2 villi here and only 3 tuft cells. So they're less than 1% of the cells really. But if you infect this mouse with a hookworm for 7 days, now you have this incredible increase in the number of tuft cells, a tuft cell hyperplasia that develops. And the reason this is possible is because of the way that the intestinal epithelium works. You have your large villi and then the smaller, more numerous crypts down below. The stem cells reside at the base of the crypts there, and they are incredibly prolific, replacing the entire lining of your intestine every 5 days or so. And as the cells emerge out of the crypts, they adopt their terminal lineages that is mostly absorptive enterocytes, but then there are less frequent secretory lineages and also the tuft cells. And what we worked out at the time, and we called it the Tuft ILC2 circuit, is that this is a feed-forward effect where in the presence of worms in the lumen, tuft cells release IL-25. That activates these innate lymphoid cells. They make canonical type 2 cytokines that do all kinds of things like act on eosinophils and mast cells. But additionally, this IL-13 that the ILC2s are making signals back to epithelial cells and drives the production of tuft cells, so that as you're replacing the lining of your intestine in the presence of IL-13, you develop tuft cell hyperplasia. And so tuft cells are essentially promoting their own production. So we discovered that together with Philippe Jay's lab in France, and then right around that same time, Michael Howard and Wendy Garrett's lab discovered the exact same thing, but came from a totally different direction. He was interested in these protists that turns out reside in many mouse strains in vivariums or all around the world and in the wild. And it turns out that when these are in the lumen, they trigger this exact same circuit. So tuft cells, IL-25, ILC2s, more tuft cells. And so all in all, this was really exciting step forward because it was really defining a missing link in type 2 immunity. Here's the cell that is perfectly positioned to monitor what's happening in the lumen of the small intestine, and then equipped to talk to immune cells and regulate the immune response downstream. And if you break this circuit in any way, like you take away IL-25 or you take away ILC2s or you take away IL-13 signaling in the epithelium, you get a big defect in the anti-worm immune response. Okay, so since that time and since starting my lab, we've really been trying to fill in some of the gaps here and expand this model and understand how the sensing is working, what else tuft cells are doing, how this differentiation works. And I'll just highlight a few of our findings quickly. The first one was that we identified how tuft cells sense these protists, and that's because tuft cells have a receptor for succinate, a G protein coupled receptor on their surface, and it can signal through this channel TRPM5 that's really critical for the function of tuft cells and also taste cells. Depolarizes the cell, and we think it drives effector release. So under anaerobic conditions, a number of microbes, including the protists and certain bacteria, will secrete succinate as a metabolite, and the tuft cells can sense that and turn on this circuit. And in fact, if you just take succinate and put it in the drinking water of the mice for a week, and then go back, their small intestine looks essentially like they have a worm infection. So you get a bona fide type 2 immune response happening there. Interestingly, this succinate receptor, while it's required for sensing these microbes, is completely dispensable for sensing of worms. We still don't know how tuft cells detect the presence of worms. That's something we're working on. But it sort of uncovers this idea that tuft cells are in part monitoring microbial metabolism, essentially, and they can sense when that metabolism is out of balance and there's too much succinate being produced. Why that's coupled to a type 2 immune response remains a little unclear. Okay, another thing that we described, and this was work done by one of the first graduate students in the lab, Jack McGinty, is that tuft cells, in addition to making IL-25, also make a lipid called leukotriene C4, part of the cysteinoleukotriene family. And these two together give you the optimal activation of ILC2s. They essentially replicate what TCR signaling does for a T cell in this innate cell. And it turns out if tuft cells are unable to make these leukotrienes during a worm infection, you have a delayed activation of the circuit and delayed clearance. But interestingly, this is completely dispensable for the succinate pathway. So there's again this split between the succinate sensing of microbes and the sensing of helminths, where the kind of the signals and effectors that regulate it are different. Okay, so one of the things that remained unresolved for us was we didn't understand why this happens, because most of the functions that we had discovered for tuft cells identified them as sentinels, as cells that were sitting there monitoring the lumen and then raising the alarm when they detected something. But why would you make more sentinels if you've already detected the thing? So we always thought there had to be some additional effector function that this tuft cell hyperplasia was contributing to the immune response. And so the work I'm going to show you, we think might start to answer this question. And this is work that's been done by Tyler Billop. He's another graduate student in the lab. This is on bioRxiv, and it's in press at Immunity, and hopefully will be published soon. Okay, so Tyler was interested in the fact that tuft cells also can make acetylcholine. This was known for tuft cells in multiple locations, including the intestine, but hadn't really been quantified carefully in the intestine. So we combined a tuft cell reporter, the FLAIR25 mice, with a chat reporter. Chat is the enzyme that synthesizes acetylcholine. And if you look in the small intestinal epithelium, you find rare chat positive cells, and every one of those is a tuft cell. So they are the only cells that can make acetylcholine in the small intestinal epithelium. But the converse is not true. So not all of the tuft cells are chat positive. And it's actually this, you find a gradient where it's about 50-50 and right near the stomach. And as you move down towards the cecum, it's closer to 90% of the tuft cells are chat positive. And we spent a lot of time trying to figure out whether these are like two subsets of tuft cells and how it's regulated. And I'll just say we didn't figure it out. We don't know, we don't really understand it. Okay, but really the question was a mechanistic one. Why are tuft cells making acetylcholine in the small intestine? And when we started this, there was a whole lot of data on acetylcholine production by tuft cells in the airways, and all of it sort of, and also in the bladder. And it converged on this idea that the tuft cell acetylcholine was being used to signal to nearby neurons to drive things like inflammation or regulate breathing or initiate urination. And I'll just say right now that that is entirely possible, that in the intestine, tuft cells are also talking to the enteric neurons. It is not, however, what we focused on in this project. Instead, we thought a lot about the function of acetylcholine in the small intestine, and it's really well known as a secretagogue. Almost all epithelial cells express muscarinic acetylcholine receptors, and signaling through them acutely drives secretory processes. So in enterocytes, this signaling leads to a calcium influx that opens a chloride channel. And when chloride leaves the cell, this is the mechanism for secreting water, right? It's the fundamental process of a mucosal barrier, is to hydrate the mucus and otherwise to kind of flush that barrier. And classically, it's always been either assumed or demonstrated that the acetylcholine doing this is coming from enteric neurons. But Tyler just wanted to ask the question whether there are scenarios in which actually maybe it's the tuft cells that make the acetylcholine. The way that Tyler did this is he took advantage of this thing called an Ussing chamber. This is something that physiologists have been using for a long time. It allows you to mount a piece of epithelium, so in our case, a small piece of intestine, between two physiologically buffered chambers, voltage clamp the whole thing, and thereby measure ion movement across that epithelium. And that's measured by the current you need to inject to maintain the voltage. It's also called the ISC, or induced short circuit current. So this is what happens when Tyler mounts a little piece of small intestine in an Ussing chamber and then stimulates the apical, so the luminal side of that epithelium with succinate, right? Which was the only ligand that we knew of for intestinal tuft cells. And you see very rapidly, within seconds, you get this increase in the ISC, which means an increased movement of ions across the epithelium. And you can quantify that as the delta ISC over here. And you'll see that over and over again in the next slides. And this lasts only a few minutes. It's a pretty short response, but it's very similar in shape to the response you get if you use an acetylcholine mimic, this carbachol here. Now, one thing is that the succinate we were using was complexed with sodium, and we did see that sodium chloride alone induced some changes in ion. So to eliminate that effect, many of the experiments I'm going to show you, actually, we use this synthetic succinate receptor agonist called cis-epoxy succinic acid, or CESA, that it eliminates that sodium component. Okay, so the thing about the Ussing chamber is that it doesn't care what ion is moving. It could be negative ions going out or positive ions coming in. And so the first thing we wanted to do was convince ourselves that what we're mostly measuring here is chloride. And Tyler did that a number of ways. One was to just remove the chloride from the media. You can replace it with gluconate. And when he did that, that response to this the succinate receptor agonist CESA, that goes away. And it also inhibits the response to carbachol, which is expected. The other thing he did is he used bumetanide to block the uptake of chloride, the basolateral uptake of chloride. In order for epithelial cells to continue to secrete chloride, they need to replenish their stores basolaterally, and bumetanide will block that. And that, again, almost wiped out the response to CESA and decreased the response to carbachol. So we think what we're measuring here is, in fact, mostly chloride, which implies that there's also water being secreted, which I'll come back to later. And so at that point, then we could use the tools that we had in the lab, which consisted of a lot of genetic mouse models that perturb tuft cells. So you can see that mice that don't have the succinate receptor, or mice that don't have this downstream channel, the TRPM5, or mice that don't have tuft cells at all because they're POU2F3 knockouts, they all fail to respond to CESA, but the response to carbachol is intact as expected. It just is a nice positive confirmation that the tissue is still capable of responding, but just not to this tuft cell ligand. And additionally, if we delete chat, so again, that's the enzyme that makes acetylcholine. If we delete it only from tuft cells using a POU2F3 Cre-ERT2 with a floxed allele, we eliminate the response to CSA, but do nothing to the response to carbachol. So indeed, this seems to be chemosensing by tuft cells that is turned into this ion flux via the production of acetylcholine. Tyler did a whole lot of other experiments to kind of rule out other things that might be involved. So for example, he found that the succinate-induced ion flux does not require enteric neurons in any way. He did that by ablating neurons, by inhibiting neurons. So we're quite confident about that. There's no role for mast cells. There's no role for substance P, which also is known to drive ion flux, and no role for nicotinic acetylcholine receptors. What is required is muscarinic acetylcholine receptors, and you can block those specifically with atropine. And so now when Tyler uses atropine, he can completely block the CESA response and, as expected, completely block the carbachol response, because this is the receptor that carbachol signals through. And the other thing that the Ussing chamber allowed him to do is to show that the response to the CESA is coming from apical sensing, but that the acetylcholine acts basally. So when you block it basally, you can wipe that out, but not if you block it apically or luminally. Okay, so at that point, we wanted to think about other tissues. Is this just a phenomenon that happens in the intestine? No, it's not. So you can see exactly the same thing in the trachea, where there are also tuft cells and they also express the succinate receptor. You can see a response to succinate that is eliminated by the loss of the succinate receptor, TRPM5 or POU2F3. But it does not happen in other intestinal sites. So in the cecum, right next to the ileum, where Tyler was initially doing all this work, you do not see any tuft cell dependent response to succinate. Same is true in the colon, and same is even true in the proximal small intestine. So it turns out that in the intestinal tract, this response is really restricted to the distal. The response to succinate is restricted to the distal small intestine. And that's actually in line with other things we've seen, including the fact that tuft cells in the colon don't express the succinate receptor, and tuft cells in the proximal small intestine also express much, much less of it. So it's determined basically by where the receptor is expressed. And so we needed a way to work around that. How could we stimulate tuft cells in these other sites so that we could ask whether they can still regulate the ion flux even if they don't respond to succinate? And right, so at that point, we were really lucky that right around this time, there was a publication of work that was led by Richard Myers at Takeda, and they had done a screen to identify TRPM5 agonists. Now, TRPM5, again, is this channel that's downstream of the receptor and we think is really central to all of the effector functions of tuft cells. And so they had identified a bunch of lead compounds, including one that they just called compound 8, that was one of the most potent. And they shared that with us so that we could try to use it as a tool to activate tuft cells. And it works, so that was really nice. So now we can stimulate this ISC response in the proximal small intestine, and it requires TRPM5. There's a bit of a non-specific slow response here, but the rapid response is all TRPM5 dependent. And it works in the distal small intestine too, and it also works in the colon. So we can use this class 8 to induce ion flux in the colon, and it requires the presence of tuft cells. So the thing is that I mentioned that we think we're measuring the movement of chloride, and that would imply that fluid is also going out. And we wanted to see if we could measure that. And for this, we were really lucky to find out that Connie Fung, a postdoc in Michael Howitt's lab at Stanford, had actually already been working with class 8. They had started a collaboration with Takeda before us, and they had done some of this in vivo work. And they had found that when they gavaged class 8 into mice, what they could see is that this led to increased fecal water content. Or really what's happening here is they always started this in the morning, and which is the time when mice stop drinking. And so in the vehicle treated mice, you see that their fecal water content decreases because they are drinking less, whereas you maintain that fecal water content when you gavage with class 8. And the really important thing is that this requires a chat in the epithelium, so tuft cells, right? So you lose this effect of the class 8 gavage in mice that don't have chat in the epithelium. So we think that indeed activating tuft cells leads to production of fluid, release of fluid from the intestinal epithelium. And this is a model summarizing what I told you. It's sort of defining this epithelium intrinsic response unit where you take the sensing function of the tuft cell and you couple it with the effector function, the secretory function of its neighboring cells, driving chloride out and water that follows that. And I will also mention that since we started this project, which was at this point quite a while ago, other people have also expanded on this idea that tuft cells are not just signaling to cells in the underlying tissue, but actually signaling to neighboring epithelial cells. So the first paper that really described that was this one from Wolfgang Kummer's group, showing that acetylcholine in the airways, acetylcholine from tuft cells can drive the mucociliary clearance mechanisms of neighboring ciliated cells. That same group also showed that in the gallbladder, acetylcholine from tuft cells could drive mucus release from neighboring cholangiocytes. And very recently, that again, the same group, again in the airway, showed this response to succinate in the airway, where activating tuft cells led to activation of neighboring epithelial cells. But what none of these papers addressed, and which was really our reason for studying this in the first place, was what does all of this have to do with type 2 immunity and Helminth infection? And so to explain that, let me explain how our understanding of how the immune system gets rid of a worm in the intestine. And it's really a two-step process. So the first step is a priming process that prepares the tissue for the expulsion event. There's a whole lot of remodeling and activation that has to happen. You need to change the epithelial composition. You need to increase mucus. You need to change the contractility of the intestine, all these things. And so this is driven basically by the Tuft ILC2 circuit and the production of type 2 cytokines. And in this particular model, that happens these worms actually passage through the lung before they get to the intestine, but from about day 2 to day 6 in the intestine, you're priming the tissue. And then there's an acute event that happens around day 7 that expels the worms out of the mouse. So this is this particular model does not, it's not a chronic infection, it's expelled. And that's sometimes called weep and sweep, where the weep is the production of fluid and mucus to detach, dislodge the worms. And the sweep is the hypercontraction of the smooth muscle surrounding the intestine to push the worms out and expel them. And what's notable is that you can link acetylcholine directly to all of these things, right? So acetylcholine signaling on smooth muscle, it will contract it. Acetylcholine signaling on goblet cells will cause mucus secretion, and acetylcholine signaling on enterocytes will cause fluid secretion. And in fact, the worms don't like this. So for decades, we've known that worms secrete a whole lot of acetylcholine esterases, presumably to counteract the host's acetylcholine-mediated immune effects. Okay, so can we link this to what we were studying in tuft cells? So Tyler again took these mice that lacked chat just in either tuft cells or just in the epithelium, but again, the only cells in the epithelium that express chat are tuft cells. And he asked whether they had any defect in clearing the worms, and they do. So the worms colonize the intestine normally by day 5, but then in the whereas in the Cre-negative mice, they're mostly gone by day 7. They persist in the Cre-positive mice until day 7 and even day 9, although by day 14 they are cleared, which is very similar to what happens if you eliminate other tuft cell effector molecules. Okay, so does this have anything to do with that priming and the activation of the circuit? No, any way that we can measure the function of this circuit with and without tuft cell chat, there's no difference. So the activation of ILC2s, measuring their IL-13 production across multiple time points, is unchanged whether or not tuft cells have chat. So they don't seem to be using acetylcholine to talk to ILC2s. And the whole remodeling that happens, the tuft cell hyperplasia, the increase in goblet cells, there's actually a lengthening of the whole intestine that happens. That all still occurs normally whether or not tuft cells have chat. So we think that the priming, that day 2 to day 6 event is functioning normally. And to further prove that, Tyler actually did an experiment where he waited until 5 days after infection to delete chat, and then looked on day 8 at clearance, and he still saw a defect, really saying you need tuft cell chat in those final days when you have acute clearance of the worms. Okay, so that says it's probably involved in weep and sweep. So which is it? And this is where we were lucky to collaborate with Tran Wu, because he had a setup in the lab that would allow him to ask the question whether activating tuft cells would acutely activate contractility of the intestine. So mounting a piece of intestine in an organ bath and then stimulating it and tracking contraction over time. And what you can see is that when stimulated, so first of all, we gave these mice IL-25, so they would have lots of tuft cells in the intestine, and then stimulated with class 8, and you see no change. There's no change in the number of peaks or in the amplitude of the peaks. Whereas if you give the positive control carbachol, you see this acute response. Similar to this, Tyler did a transit experiment and just looking how far a dye would move through the small intestine in 30 minutes. And again, it didn't seem to matter whether or not there was acetylcholine in the tuft cells. So we haven't been able to really link tuft cell acetylcholine to this contraction, to the sweep mechanism. And so we focused instead on the weeping and whether the fluid secretion and the ion flux we were measuring was increased during type 2 immunity. And it is. You can see that as you progress through an infection from day 0 to day 7, that ISC in response to succinate increases during that time. But you might notice that this increase is not as big as you would expect, right? I showed you there was like a 10-fold increase in the number of tuft cells. And we think the reason for that, there's a few reasons. One is that actually, and this was known previously, the total response to carbachol is reduced as inflammation occurs. And the other thing is that not all of the tuft cells that emerge during hyperplasia are chat positive. So the total numbers of CHAP-positive tuft cells still increases, but it doesn't increase as much as the total as all tuft cells. And so for these reasons, it's not as big an increase as the hyperplasia itself, but it's still very clearly increasing with inflammation. And the way that we're thinking about this is that because acetylcholine generally does not signal very far once it's outside of a cell, there's a lot of acetylcholine esterases in the extracellular environment. And so we think that every tuft cell basically can reach a ring of, say, maybe 10 neighboring epithelial cells. And by increasing the number of tuft cells, you're basically increasing the coverage, the amount of the epithelium that you're able to reach with this effector response, and decreasing the hiding places, the potential hiding places for the worm. And thereby, wherever the worm tries to go, you're gonna engage this effector response that we think can help clear the worm out of the intestine. Okay, so summarizing this first part, then, what Tyler uncovered is this acetylcholine-dependent regulation of epithelial ion flux. And this, of course, supports basic mucosal barrier function. You need to hydrate your mucosal barrier in order for it to function. And we think it contributes to the weeping that helps expel worms, together with lots and lots of other things that are happening. This is a complex process. And the other thing that it uncovers is really the way that tuft cells function across time. So you have the acetylcholine-driven response that occurs in seconds or minutes. It's really an acute response. At the same time, you have the ILC2, tuft ILC2 circuit that activates over the course of days and leads to the remodeling that really positions this acute response to help with the worm clearance. And so that's sort of a biphasic thing. And as a transition to the next thing I'm going to talk about, what's missing here is what do tuft cells do on an even longer time scale, weeks and months, when you have, for example, chronic worm infection or reinfection. And these are the kinds of things that are much more common in humans. We tend to be chronically infected by the worms that reside in our intestine. We have a lot of deworming programs that'll eliminate worms, but almost always people are then reinfected later. So those were the kinds of things that postdoc Alo Stanbury wanted to study, and just very generally to start to think about what do tuft cells do in the context of adaptive immunity. Everything that's been published about tuft cells to date is really about acute and innate kinds of things. So how do these cells engage with adaptive immunity? Okay, and to do this, we had to switch worm models because the Snipostrongylis hookworm model, again, is cleared within 7 days. And we switched to H. polygyrus. This is a worm that infects directly via the oral route. The infectious larvae enter the small intestine. They immediately burrow through the epithelium and insist in the tissue below the epithelium, where they form, there's an immune response that forms a granuloma, and they mature into adults. And starting around day 6, they leave the granuloma, migrate back out through the epithelium, and take up residence in the lumen. And this is actually a chronic infection in mice. It'll last for months. It takes a very long time to eliminate these worms. However, you can drug clear them after 2 weeks or so, wait, say 4 weeks for everything to die down and memory to establish, and then infect again with a secondary infection. And this time you have a memory response that eliminates these worms by day 14 in a wild type mouse. And it's known that T cells are really critical. CD4, Th2 T cells are really critical for this memory response. And it's been thought that it's actually this larger granuloma that's really key in killing and restricting the worms. Although I'll show you that we're not sure that's really all of the story. Okay, so this is the model we used. Another way to show it is that you have a primary infection, you wait 2 weeks to clear it, you wait another 4 to 5 weeks for memory, and then you infect again, and you can do analysis days post-secondary infection. And some of the questions that Alo was asking was, one, how is this tuft cell hyperplasia regulated in a chronic or a secondary infection? And in particular, the question is, what's the relative contribution of ILC2s and Th2s? And this has been a question that's been a little hard to answer in the field because we don't have good tools to selectively manipulate ILC2s and Th2s. But I'll show you we have something that helps to do that now. And then the other question really was, do you even need tuft cells once you have T cells? Because ILC2s have no way to detect the worm alone. They need tuft cells to act as their eyes and to tell them that there's a worm there and to activate the ILC2s. T cells, on the other hand, have a TCR. They can bind to helminth-derived antigen directly, and therefore you could imagine that you wouldn't need the sentinel function of the tuft cells at all. So that was another question that we had. Okay, and I'm gonna, this is all still work in progress, and I'm gonna summarize it pretty quickly for the sake of time. Although I'm happy to answer additional questions. So one of the tools that we used here is that we were really lucky to get these mice from Golnaz Vahedi and Jorge Henao Mejia. They sent them before they were even published, although now they're described in this Nature Immunology study. And what they did is what Golnaz does really well is to study the 3D interactions of DNA, of the genome. And they did this in type 2 cells and in ILCs, and they were able to identify this distal enhancer of ID2. You would never have realized that this is interacting with the ID2 promoter until you do that, but it turns out in 3D space, it folds in and it does. And it turns out it's only open and active, this LCR1 site, only open and active in ILC2s. And so they made a knockout mouse, an LCR1 knockout mouse, in which you can prime Th2s fine. You don't need this at all to make a Th2. ILC1s and ILC3s are fine, but ILC2s are severely blunted. They're way down in all tissues and sometimes even nearly absent. So this is a nice sort of ILC2 knockout mouse that they shared with us. And so when Allo started to look in this at this secondary infection, she found that, kind of as you would expect, T cells completely take over. So after a primary infection, 2 days after, if you look at who's making IL-13, it's all ILC2s, as you would expect. During memory, everything turns off. Nobody's making IL-13. And then when you infect again, during that secondary infection and ask who's making IL-13, now it's all T cells doing it, and they vastly outnumber the ILC2s. And they're become totally the dominant source of IL-13. And they're also become functionally the important cell. So if you look at tuft cell hyperplasia 4 days after secondary infection, you do not need ILC2s to get tuft cell hyperplasia. This is totally different than what I showed you earlier, right? In Nippo, it's all ILC2 dependent. Now it's ILC2 independent, but it's T cell dependent. So if we deplete T cells with anti-CD4, you lose most of the tuft cell hyperplasia. And what's interesting is that it's, despite being T cell regulated, it's still tissue intrinsic. So you do not, if we block the recruitment of additional T cells using FTY720, you still get totally normal tuft cell hyperplasia occurring in the tissue. And in fact, if you look at worm clearance, here's a primary infection. So we put in about 200 worms and we get about 150. This represents the lack of clearance in a primary. Secondary infection in a wild type mouse, you clear the worms. Without T cells, without CD4s, you don't. That was known already. But what we found additionally is that if you block recruitment of new cells from the blood, you still clear the worms essentially just as well. So it has really become this tissue intrinsic circuit, just like it was before, but before it was ILC2 driven, it's now Th2 driven. And so these Th2s have really functionally replaced the ILC2s in the small intestine. Okay, so that's maybe not that surprising. This is adaptive immunity, right? This is what you would expect. T cells take over. But what about that second question? Now that the T cells are there and they can get antigen directly from the helminth and they can regulate this granuloma, which they're known to do you still need tuft cells, or do they become dispensable? The answer is yes, you still absolutely do need tuft cells. So again, here's the primary infection. With or without tuft cells doesn't make a difference. Nobody's clearing worms. Wild type mice clear in the secondary infection, but mice that don't have tuft cells or that don't have that TRPM5 effector channel cannot clear any of the worms at all. And the same is true if the mice don't have IL-25 or if they can't make leukotrienes. So these tuft cell effector functions are required. You can just briefly, as a side note, minimal role for IL-33, which signals through SD2, and again, little to no role for ILC2s in this secondary response. Okay, so these are all constitutive knockouts, so they lack tuft cells both during the primary infection and during the secondary. So when do you need the tuft cells? So we did time deletions. Here we are driving expression of DTA with a tuft cell Cree and tamoxifen. And so we allow everything all the way until the secondary infection. Everything happens with no deletion, wild type mice essentially. And then right before and during the secondary infection, we delete tuft cells. And you can see that if we delete them throughout the entire secondary infection, we again get this delay, this defect in worm clearance. And even if we wait until halfway through and we only delete tuft cells in the last week of secondary infection, we still get a defect in clearance. And that's interesting because that whole granuloma phase of the infection that I told you about, that all happens in the first week. But after that, the worms would be out of the granuloma. And in fact, if we delete tuft cells, it has no effect on the number of granulomas that we can find and their size. So it seems that in this context, granuloma formation is normal, and yet you fail to clear the worms out of the intestine. And it really emphasizes that tuft cells are doing something they have additional effector functions that are important in clearance of this chronic or secondary infection. What are they doing? And this is part of what we're still really working on. But one area we've focused on is again on the T cells, looking back at the T cells. And it turns out that even though H. poli only lives in the intestine, you can find memory Th2 cells in many organs. And so Allo sorted them out of all of these organs, 4 weeks after clearing infection. And what was striking is that if you look at some of the receptors on these cells, the ones that are in the small intestinal lamina propria are the only ones really, and some in the mesenteric lymph node, that express high levels of IL-17RB, which is part of the IL-25 receptor. And all of these memory cells express cysteinoleukotriene receptor 1, which is the receptor for LTC4. So the T cells that are in the small intestine are uniquely set up to get signals from tuft cells, IL-25 and leukotriene C4. And so, in fact, if we make memory mice and sort them out of the small intestine into a dish, we can activate them by giving them just IL-25 or just leukotrienes in the complete absence of antigen. And if we combine the two, it's the best. And we're starting to approach what happens with PMA ionomycin, right? That's the best way to activate a T cell. We get similar kind of activation without antigen here. And the same is true if we make memory cells and then we either reinfect the mouse or we just inject the mouse with a recombinant IL-25, we can get similar reactivation of the T cells. The production of IL-13 is essentially the same. And so there's a real capacity for these cells to be activated without antigen by tuft cell effectors. And conversely, if we delete tuft cells and then look at the activation of the T cells after secondary infection, it's decreased. It's not gone completely, right? And that makes sense. These cells do still have a T cell receptor and they can still get antigen, but they do seem to require some signals from the tuft cells. Okay, so that's where we are. And so to kind of summarize, I told you about our earlier work looking at succinate and leukotrienes, but then I emphasized this new study where we've really focused on tuft cells making acetylcholine and how that talks to neighboring epithelial cells. And then also the way that as you move into chronic or secondary infection, Th2s really come in, and this becomes rewired, we think, into a tuft Th2 circuit, essentially, where the Th2s are both driving the tuft cell hyperplasia and getting signals from the tuft cells. And the implications of this are that we think tuft cells are a really interesting therapeutic target if you want to modulate intestinal physiology or the immune state. If you had a way to turn tuft cells up or down, that gives you access to immune and physiologic pathways in the intestine. Also, our work highlights that if you want to make a successful vaccine against helminths, which is kind of the holy grail, you're going to need to find a way to establish tissue-resident Th2s. Those are the key cells that are going to be driving clearance. And then lastly, if it's really true that these Th2s can be activated without antigen, that has interesting implications because maybe they can be engaged in defense against a heterologous infection, where you don't have matching antigen. Or they might also be contributing to pathology and allergic disease. Once you've established them, the tuft cells might amplify them. But then I'll also say we're also thinking about whether there are other cellular targets of tuft cells and the effectors they make that might be important here. Okay, so that's everything. And I'll just end by saying a big thank you to the people in the lab. I've had just outstanding trainees who you see have done amazing work. I highlighted the work done by Tyler Billup today and by Alison Stanberry. We've had great funding and great collaborators for that acetylcholine paper, really critical contributions from Michael Howitt and from Tron and Jason Debley, who actually helped us start the project. So it's great to work in a supportive scientific community that expands outside of Seattle, and great that we have this venue to kind of talk to people anywhere. And I'm happy to take questions.